So today I'm going to talk about just sort of foundation models in general and what they are, and a lot of the way that I think about training foundation models, mostly on biological data, but for contexts like astrobiology, and like very sort of weird environmental contexts where your place where you're trying to deploy your model looks very different than the training data that you may have for training your actual foundation model. If I can advance slides, there we go. So I'm at the University of Georgia in the Marine Sciences Department, and I've always been sort of a marine microbiologist by training, and I study microbes that impact carbon cycling and try and map that back onto their environmental conditions and how those co-influence one another and what that means for sort of global scale carbon cycling. And so I'm not going to talk very much about my research, but the main notes I want to hit here is that, everywhere you go in the ocean's not so different from space in many ways, and that it's very expensive to get to. It's very time consuming to get to. And so when you go out there and you spend all this money trying to gather as much data as you can, it's very precious data. But it also tends to be very limited data because of the nature of having to go out on a boat and collect that data. So, when we go out there, we encounter new microbes everywhere we go. They have hugely different functional capabilities in different locations and different environments. Everywhere we look, we're finding something new. And that makes it very challenging to predict what microbes are where and what they're doing and how that impacts, global scale carbon cycling and these, planetary scale processes that we care about. So marine microbes, they're these very micro scale processes, these molecular scale processes that add up to ecosystem scale processes and global scale processes, and over time, shape the habitability of a planet, right? And also have been shaping sort of humankind and how we use biology, both on Earth, of course, and there's a huge burgeoning or huge, thriving biotech market, but also in, as we try and travel into space in terms of um, making the space environment habitable for humans, so we can explore all of the planets we want to explore, right? And so when we're looking for new biological information, we want to discuss something new. Most biology that we know of is in these environmental microbes, and they're in these environmental microbes that are in the rare environments, the weird environments that are hard and expensive to access, and that are, because of all those things, represented by these really small, precious data sets. And so, we have two ways of going about trying to understand these systems. We can try and take the data that we have and do some computational predictions. And then you also have to do some experimental screening, right? And understanding what's actually going on there and verifying your computational predictions. But when you're trying to predict these really complex systems, turns out that that's pretty hard, right? And that's partially because they're really complex. But it's also, especially in the case of biology, but many other um, data modalities as well, something that I like to call the microbial dark matter of it all. So in the case of biological data, and most of the stuff that I'll talk about today is very biased towards biological data because that's what I work on and that's what I like, but I think can be sort of translated to other data modalities that we care about for astrobiology. But when I talk about microbial dark matter, what I mean is that when you go into the environment, you take a sample, we have sequencing technologies, right? We can sequence everything that's there. But we're very bad at telling you what those sequences actually mean. And that's because, we can't get microbes in culture, right? Things that are living in the environment are living in very different conditions than we tend to grow them in the lab. And so, less than 1% of microbes can be cultured. There are entire phyla of microbes that we don't have a single cultured representative for. So that's these black bars here. You can see in some of this is just a list of microbial phyla, and we have cultured representatives for some rather small fraction of them at the phylum level. That's a pretty big taxonomic category, right? And so what we tend to try and do is we take these sequencing reads that we can get pretty cheaply and easily these days, and we try and use computational approaches to figure out, who's there, what function are they doing. And conventionally, in bioinformatics, you take these reads, maybe you assemble them, maybe you don't, and then you align them to a reference database that we have of all the microbes and their genomes that we know of, or all of the genes and any functions we know of what those genes do. And then we try and say, like, okay, if this sequencing read looks like this thing in the reference database, we're going to say it has the same function or it's from the same bug. The issue is that it's entirely dependent on what you have in your reference database. And so your reference database in biology is extremely biased and also very wrong. So in the case of the ocean, where I do most of my work, 30% of your environmental sample, when you databases, will map to anything in a reference database that has any kind of function that you know of. Most of those are functions that have not been shown in the lab to actually do that function. So it's been based off of homology and things. So if you're actually just comparing your read to genes that have experimental annotation, we know it does this function, like 1% of your reads will map to one of those genes. So we are mostly in the dark in terms of knowing what function microbes are doing in the environment. And that's because a lot of our reference databases have this really high rate of misannotation. And I won't go into why, but it's basically because of the way that we do homology-based bioinformatics inference. So we're really reliant with our sort of conventional methods on these reference databases, on the things that are in the realm of the known, right? There are sort of known knowns. And so that's really limiting if you're in a field like environmental microbiology in general or astrobiology, where you have really high dimensional, really complex things that you're trying to understand. You have really low sample sizes, and the things that you are measuring are really poorly represented in the sort of analogous environments that you're trying to compare it to. And so I think that's a really key blocker to certainly conventional analytical methods, but it's also a real blocker in training your AI models. So I'll talk about why that is. And you need sort of special considerations to sort of sidestep this issue. But I think foundation models are a real key enabler to us being able to better capture these sort of novel environments in biology and these novel environments or the potential. So what is a foundation model? So I'll give sort of the example of large language models because I think that's sort of the foundation models most of us interact with on a daily basis, right? And it's also pretty analogous to biological systems for reasons you'll see why. So a large language model like GPT-4 or Claude or Gemini, any of these, are models that are trained on a massive amount of data on a very simple task. So in all these cases, they have, language that they've scraped from the internet that is pretty representative of all of human language, right? And they say, okay, here's a sequence. I'm going to mask out the last word in the sequence, and I'm going to ask the model to fill in what that word should be. And so the model learns over time, what other words to pay attention to in this sentence that informs what word might fit here given the context of the language around it. And so from that, even though it's not doing anything really that useful yet, right? It's learning sort of the rules of grammar. It's learning how people talk. It's learning a lot of sort of fundamental features of language that can then be leveraged to do all sorts of downstream things. And so, I'm sure most of you have interacted with some kind of chatbot in the recent history, maybe right now. And so what you can basically do from this basic, very, very basic training task that you have basically infinite training data, right? Because you can create your labels from the input itself. You do this self-supervised training. You can then fine tune it on fairly limited training data downstream to do all sorts of useful things. So given that really simple sort of self-supervised learning objective, you can then do lots of things like that we call in-context learning. So, I ask it a question, it gives me an answer, following instructions, summarization, classification. You can fine-tune this large language model to do all sorts of useful things with relatively little data. So the reason why this is, I want to back up a little bit to talk about just the idea behind foundation models, and then how we then apply it to these downstream things. So, when we're trying to model complex systems, we need a complex model to model that complex system. But the more complex your model is that you need to train, the more data you need to train it. And so if you're in a situation like we have in environmental microbiology, like we have in astrobiology, where you have really small data sets, you need to find a way to get that big data performance on your very small sort of labeled data set that you've painstakingly collected, right? So most biological data is this small data here. So we're kind of on this small data. If you try and train a model from scratch on this small data set, it doesn't matter how big your model is, you're not going to get that much better performance because the data you have just doesn't, you don't have enough data to sort of to learn some of these fundamental features of your underlying data modality, right? So in foundation models, if you can find a source of data from the same data modality, you can start training these much bigger, deeper models where you learn these fundamental features underlying, biological data, sequence data, fill in your data modality here, and then use it to get that, to learn those fundamental features underlying your data, and then take your small data set, not have to learn those features again, um, and be able to fine tune it to get that deep model performance on what is actually small data. And that's the whole idea behind transfer learning. So if you're, so to sort of describe how this works, if you take your small data set and you try and train it from scratch to predict the thing you actually care about, right? You just don't have very much data. It's not going to do very well, right? Foundation model that you've trained on a massive amount of data on an unrelated learning task. It almost doesn't matter what that learning task is, just something that's going to inform the model about the features underlying that data modality. You can then learn this domain knowledge, the fundamental features that underlie all of biology. We don't need to relearn on every data set what a codon is, right? We could probably learn that from a different task on a larger amount of data. You then transfer that sort of fundamental knowledge over to your new model, preload those weights, and then fine tune it on your small data set and get much, much better performance because you don't have to relearn all of those basic things about biology. You already know those things. And so the reason why this works, I like this example from imagery because I think it really illustrates this well, is that deep learning models learn complex functions successfully from universal to task-specific features. So if you have a deep model that's 12 layers long, you're as you put input, say, an image, you're going to learn sort of more complex features about images as you go successfully down those 12 layers. So in the first layer, you might be learning things like edges, right? This is basically an edge detector. Your layer 2, now you're starting to put them together into, corners and stripes and sort of basic geometric patterns. Layer 3, you're getting more, complex patterns. And then it's only in the later layers where you start to recognize the very specific thing you're trying to classify. I can see a dog face, I can see bird legs specifically. Like this layer is for detecting that level of specificity. And so then when you go and you fine tune your model on your new data set, say you're trying to predict whether this cell has a tumor or not, you can port this model over, basically chop off these last layers, and retrain them. but you don't have to relearn like what's a curve, what's an edge, what's a, reflectance of this light, right? You've already learned that. So you don't need to spend your precious data information that you've gathered for this tumor data set on relearning those features. You can learn, the specific things related to whether there's a tumor in this cell or not, even though there's nothing even looking like this cell in the original training data set. And so in biology, we have a lot of these sort of analogous models that have come out now. I'm giving just a couple examples of the popular ones. You may have heard of AlphaFold or ESM2. And my sort of hobby horse, my mission in science is to change how we approach training these biological foundation models. And I'll sort of detail why. So AlphaFold, it's a super popular model. It can take a bunch of sequences and predict the structure, the 3D structure of that protein, of that amino acid sequence. ESM2 can do similar things straight from sequence without taking some of the sort of other pre-processing that AlphaFold does. But there's a couple problems with these models. Some of them require pre-processing, so things like AlphaFold, you have to create multiple sequence alignments, things like that. They require full-length protein sequences, which means it has to be something that's in a protein reference database. And it's trying to predict things like structure, which I would argue it isn't that important to structure is a proxy for function. So why not just predict function, right? But the main thing is that these really aren't optimized for environmental settings because they're looking at they're training on basically the UniProt database or the PFAM database, things where we have really well characterized full-length proteins, often from like culture representatives. And so you have the same bias problem in the training data that these models are using. And we find that that doesn't generalize very well. And so if you want to generalize to a new environment, you need to capture in the case of biology, the biodiversity as much biodiversity as you can possibly get into your model. And so that's a lot of what we do in my lab is figuring out how to do that for biological foundation models. And so in the case of biology, that means perhaps we don't have a lot of proteins in a reference database that are representative of our environmental sequence, but we have tons and tons of environmental sequences that we've measured, right? But they're in the form of these short reads, these 100 to 150 base pair like metagenomic reads. And they're not represented in our protein databases. They're not going to map to anything in those databases. And so we train things like this looking glass model where you are training on the environmental DNA data that we have that's actually representative of the microbial tree of life, all of this microbial biodiversity that we have that isn't represented in those sort of downstream databases. And so we do something very similar to what the language models do. You're predicting a masked nucleotide from the context of the sequence around it, never mind the details. But you do this massive pre-training on DNA data from across the microbial tree of life. And from that, you can learn some of those basic functional, evolutionary, environmentally relevant features that underlie biology in general, that you can then fine tune to do all sorts of things, things like functional prediction, homology detection, environmental prediction. You can identify the importance of different nucleotides to different prediction tasks. I'm not here to talk about looking glass, but the point is that you can transfer those fundamental features to lots of, predictions about novel biology, optimal temperature, reading frame, things like that. And a big reason why this is because representing biodiversity in your models is really, really important, and I will die on this hill. So what Looking Glass does that's different than a lot of these biological foundation models is that we sample our training data from across the microbial tree of life uniformly. So we're not, taking random sequences from our reference databases, right? Because that's going to be like all E. coli genomes. We're taking DNA from microbial genomes that sort of represents this biodiversity. And then we do something really, really important for generalization, which is you split your training and validation sets according to sequence similarity. And so in this case, we basically said nothing from a genome from the same class is going to go in both the training and validation set. And what happens there is you're not going to just sort of copy memorize the sequences of your training data, right? You're going to generalize to maybe new genomes you might discover, understand some of those generalizable features. And what we find happens is you end up training a model that's more accurate with a lot less training data and training effort than if you sort of randomly sample your data. So all these blue points are randomly sampled data versus the pink, which is our actual training data. You get much higher accuracy on this self-supervised task in a lot less time. And you also capture a lot more useful, functionally relevant features. So I'm really excited about this.
Hey,
This is
Adrienne,
yeah,
can
yeah.
we just try to wrap up in about 3 minutes, please?
Yeah, sure. I
Okay,
think
thanks.
I only have a few more slides.
Okay, perfect. Thank you.
Yeah, so when you're trying to capture this biodiversity and split your data carefully to be generalizable to new environments, to new biology, you capture a lot more functionally relevant features that's then more generalizable downstream. So this is we were benchmarking Looking Glass, a much smaller model trained on much less data than a bunch of other biological foundation models. And you see much higher accuracy in things like functional prediction than a lot of these other models that are much bigger and use a lot more GPUs to train. But you see a performance hit for things like rare functional classes, which I think also gets to the problem we have in astrobiology, where we're dealing with a lot of sort of rare classes, rare situations, and so you still need those techniques alleviate but don't eliminate a lot of that training data bias. So you need to be aware of that downstream. Okay, I'm going to skip these, but the general point is that when you apply AI tools versus like conventional analytical tools, you can pull out trends in environmental samples that are, potentially different or completely reversed than your conventional tools. So this is our AI-driven tool. You can see there's a latitudinal trend here that's reversed from our conventional tools. So I think really using these AI-driven models gives us the potential to break out of this reliance on known knowns and generalize to new environments, discover things about novel biology in new environments than we could otherwise do. So I'll wrap up there. That's all I wanted to say about foundation models. I think the other thing that we work on that is of interest to this group that's worth just mentioning in one slide on my last slide is that, because all of these approaches to model training can alleviate but not eliminate that training data bias. At the end of the day, we often need to explore the data space, right, and create more experimental data. And so one of the things we're working on in the context of biology is using active machine learning to try and do things like gene functional annotation, but select which genes we annotate in an AI-driven way to sort of maximize our generalizability of our downstream functional prediction models and be able to discover new biology, fill in those data gaps, and generalize to new environmental contexts more quickly. So I think that was a very biology-centric example, but I think a lot of those things are approaches and challenges are analogous to all sorts of data that we use in astrobiology. So I'll just end on a thank you slide for all my great lab members and wrap up there.